All right, so let's review a little bit about PCR or polymerase chain reaction and what that means and how it works. We'll start with just a basic ingredients list or reagents list. So you're going to need the Taq polymerase, which doesn't denature or get destroyed in heat. We'll need primers and just individual nucleotides, the right pH, some cations to make the polymerase happy, and then DNA template that we want to copy. So how this works is we start with our template. This is kind of like a copy machine. This is going to be the first piece of paper that we put in. And this template is going to be made of DNA. So it's going to be double-stranded, one strand going 5' to 3' on the top, and the other going 5' to 3' right to left on the bottom. Okay, so that's the template. The first step is to separate those strands. So we call that denaturation. So once we separate those strands where they're denatured, the second step is to add on the primers. That's called the annealing step. So a little primer is about 20 nucleotides long, 20 bases long, and it is the reverse complement. This primer is the reverse complement of the top strand. The second primer is the reverse complement of the bottom strand. And these two primers have to be facing 5' to 3', facing towards each other, and that'll let us amplify what's in between or copy what's between them. So we've got the denature step, the annealing, the primer step. Annealing just means stick. And then the last step is to have the Taq polymerase come in and copy from that primer 5' to 3' on the top strand, and from that primer 5' to 3' on the bottom strand. So again, those primers have to be facing towards each other, and that lets us copy exactly what's in between them. So that's the first round, and we do it again in the second round. So again, denature each of these newly synthesized helices. So now we have four single-stranded pieces of DNA. The primers come in and anneal to each one of those strands, and they amplify towards each other or extend towards each other, and that's how PCR works. So it's important to remember the temperatures for these. The first step, denaturation, where you're separating the strands, that happens at about 94 or 95 degrees Celsius. The primer annealing, that needs to be a little bit lower. So that lets the primers find the exact right sequence where they're going to stick. And there's going to be millions of primers in this tube. Not nearly as much template as there are primers. So the primers are going to anneal in the 60 degrees Celsius range. And then extension, the polymerase works best at about 72 degrees. So that's going to be the best one for that. Quick side note, depending on the number of G's and C's in the template, that's going to affect the numbers of G's and C's in the primer. And the higher the number of G's and C's in the primer, the higher the annealing temperature is going to have to be. So more G's and C's would increase that temperature, less G's and C's or more A's and T's would decrease that temperature a little bit. Okay, so that's why there's a range. There is a video about this and how the process works in the tube, but remember this has to use a polymerase enzyme that is happy working at high temperatures. So they found one in a deep sea vent and they named the bacterium it came from Thermus aquaticus, and we call it TAC is the abbreviation for the bacterium, and then Taq polymerase is the enzyme they took from that bacterium. All right, so again, the primers are short. They're usually 18 to 25 bases, and they have to have this 3' hydroxyl group that's available and accessible to the polymerase so it can start adding on nucleotides going 5' to 3'. So we've got to have primers in excess, so millions of these primers floating around in the tube. That makes it more likely for a primer to come in and anneal when those strands are separated than for those two original template strands to reattach to each other. And then we do the experiment in a thermal cycler. So it's usually 35 to 40 cycles of this or rounds of PCR, and it takes usually 2 to 3 hours to get that done. Okay, and then once we're done, we'll talk about electrophoresis in a different video, but we always run a PCR product on a gel to see if it worked. If we don't get a band, then it didn't work, or we forgot to put something in the tube. And then this is going to be semi-quantitative where we can kind of see that there are brighter bands as we increase the amount of DNA that we put in each one of these wells, we're going to get brighter bands. So all we can say is that this band is brighter than that band. That means this band on the right has more DNA that was copied in it than the band on the left does, but we can't say exactly how many copies of DNA are in there. That's why it's semi-quantitative at best. Okay, so again, we've got a way now to go from one double helix to billions of double helices of that short little region we're trying to copy within just a couple of hours. So that's some pretty impressive tech.